Welcome to the lecture series on control of gene expression. I'm Janine Post of the Developmental Bioengineering Group at the University of Twente. Today I will give a general introduction to why control of gene expression is so important. Illustrations in this video are based on the book Essential Cell Biology, and this information in this lecture can be found in Chapter 8. This lecture is part of a series of 4 short lectures on gene expression control. In the first lecture, I will talk about the general principles. The central dogma of gene expression is that genes are transcribed into RNA. And the RNA is translated into protein. Ultimately, it's the proteins in a cell that dictate its function, and for a cell to obtain its functional proteins, various steps exist, each under tight control of specific proteins. Here I will show some of these control points and the gene products and proteins involved in these processes. Cells are able to change which genes they express without altering the DNA. Various experiments performed as early as the 1960s have shown that a complete new organism can be formed by taking the genome from one differentiated cell to generate a whole new organism. The principle of one of these experiments is shown in this image. The nucleus of a skin cell from an adult frog, when transplanted into an enucleated egg, once whose nucleus has been destroyed by UV light, can give rise to an entire tadpole. This shows that the nucleus from the skin cell cannot have lost any critical DNA sequences. Nuclear transplantation experiments carried out with differentiated cells taken from adult mammals, including sheep, cows, pigs, goats, and mice, have shown similar results. A nucleus removed from a differentiated cell of an adult cow can be introduced into an enucleated egg from a different cow to give rise to a calf. Different calves produced from the same differentiated cell donor are all clones of the donor, and therefore genetically identical. The cloned sheep Dolly was produced by this type of nuclear transplantation. Dolly had 3 mothers. One provided the egg, one the DNA, and a third carried the cloned embryo to term. Dolly was born in 1996 and had her first offspring, out of 6 lambs, in 1998. She later developed arthritis and progressive lung disease and was euthanized in 2003 at the age of 6.5 years. So if DNA in specialized cell types still contains the entire set of instructions to form a whole organism, it follows that the different cell types in an organism do not differ because they contain different genes, but because they express them differently. Using techniques such as mass spectrometry allows us to analyze the protein content of cells even when they are expressed in very small quantities. This enables us to generate protein expression profiles specific to different cell types. As all cell types produce proteins that are common, which we call housekeeping genes or proteins, we will always find these. These include DNA and RNA polymerases, DNA repair proteins, ribosomal RNAs, and other enzymes. Each cell type also expresses a set of genes or proteins that is unique to that cell type. Examples are hemoglobin, that is expressed in red blood cells, but also specific transcription factors that induce expression of genes specific for that cell type. One example of that is MyoD, that regulates muscle development and repair. I will get back to this later in one of the other lectures. In addition to common and cell type specific protein expression, cells also contain sets of genes and proteins that are expressed only under certain circumstances. For example, in response to extracellular cues. One example is that under starvation, intense exercise, or prolonged stress, the adrenal glands produce cortisol, which signals liver cells to boost the production of glucose, while other cells do not respond to cortisol, or can even have a different response. This indicates that due to different specialization and its corresponding gene and protein expressions, cells respond in different ways to the same extracellular signal. This is further explained in the lectures on general principles of signal transduction, which you can find on the YouTube channel. So in order for a cell to express the proteins that it needed for its function, there are different control steps that regulate the steps from gene expression to mature protein. For example, the first control point is the transcriptional control. Secondly, we have RNA processing, such as splicing, which generates variants. The RNAs that are exported to the cytoplasm are selected before they are localized to the cytoplasm or even to specific organelles in the cytoplasm. mRNAs have a certain lifetime that dictate when they are going to be degraded. mRNAs can be selected that are translated by ribosomes. We have translation control. And we also have the regulation of lifetime of the protein as well as regulation of protein activity. Like I said, that could be due to cellular signaling, which we will discuss later. However, for most genes, the main site of control is the transcriptional control. These processes will be explored in more detail in the next lecture. Thank you for watching. If you have any questions, please email me. In the next lecture, I will talk about the regulation of gene transcription.